Our second study is by Henry Weimer Skirch and Associates, 2001, Heart Rate and Energy Expenditure of Incubating Wandering Albatrosses, Basal Levels, Natural Variation, and the Effects of Human Disturbance. Albatrosses are large surface nesting birds, and like penguins, breeding pairs alternate between incubating the egg and foraging at sea. Incubating and chick brooding adults supply heat to eggs or chicks to keep them at optimal temperatures and provide protection from predators and/or environmental conditions. During incubation periods, the birds need to fast for several weeks, meaning that the bird has to have enough stored energy reserves. The rate of energy expenditure during incubation can be helpful in determining breeding success in birds like these that have to fast during prolonged incubation periods. The heart rate method, which as we went over in the first lecture, is based on the Fick equation, which demonstrates that heart rate is directly related to the rate of oxygen consumption. Heart rate can predict metabolic rate, and when combined with behavioral data, can predict energy expenditure, just like in the king penguin study. The research for this study took place during 2 austral summers as well, but only from January to March. The study was done on Possession Island, which is a small island that is part of the subarctic Crozet Archipelago, belonging to the French. Adult wandering albatrosses from colonies at Baie du Marin and Point Bas were used for this study. The great thing about these particular birds is that they had been under study since 1966, so a lot of the vital information like breeding status, age, and sex were already logged into a database. This study had 4 goals. The first was to measure heart rate and rate of oxygen consumption for resting albatrosses in a metabolic chamber and also for a separate group of birds in the field using data loggers. These 2 sets of data would be compared to determine the effect of stress on the birds in the chamber. The second goal was to examine effects of body mass, age, and sex on heart rate for birds during the incubation period. Third, the study would determine if there were variations in heart rate associated with daily rhythms and weather conditions. Fourth, the variations in heart rate caused by human disturbances would be measured to see the impact of stress on metabolic rates of incubating albatrosses. 41 birds were selected for the field study. They were a combination of both male and female. 33 of them were in the incubation period, and the other 8 were in the chick brooding period. These birds were from the Point Bas colony. This group of birds were captured while nesting, weighed, fitted with heart rate recorders, and immediately released back to their nest. For the metabolic chamber study, only 8 birds were selected. They were captured, transported in a box to the nearest base, which is about a half a kilometer. They were weighed, fitted with a heart rate recorder, and placed in a box inside of a metabolic chamber. Measurements were gathered for up to 9 hours, and then the heart rate recorders were removed and the birds were taken back to their colony and released. For this group, only non-breeding birds were chosen, so there wouldn't be any risk of them deserting nest, which is the leading cause of breeding failure. This turned out fine though, because non-breeding birds are similar in size to adult birds, and no physiological differences were expected in regard to maturity or body size. Now I'm going to go over some information about the data loggers that were used and then the metabolic chamber. I'll briefly touch down on some of the differences between birds placed in the metabolic chamber and the free-ranging birds. The heart rate loggers used on the birds were developed for human use, but the researchers adapted them for use on albatrosses. They weighed about 50 grams and were made up of 2 units, the transmitter and the receiver. The transmitters were attached to the backs of the birds using a medical-grade adhesive tape that was wrapped around several layers of feathers. 2 electrodes attached to the transmitters had to be inserted into the skin, so the insertion points were cleared of feathers and swabbed with an antiseptic before insertion. Once the transmitters were removed, the sites were coated with an antibiotic cream to prevent infection. How the loggers worked was that the transmitters would process the electric signals to separate the heartbeats and other electrical activities like muscle movement, and then they would amplify the signals and send them to the receivers. The transmitters had been previously tested on gray-lag geese for accuracy, so they were good to go. The loggers used in this study were much easier to attach and to remove than the units surgically implanted in the last study on the king penguins. Even better, it only took about 5 minutes to attach the loggers and to remove them. For the field study birds, the loggers remained attached for 24 to 36 hours and the heart rate was recorded in 60-second intervals. The time it took for the heart rate to reach basal levels was recorded. These birds were left alone for the first 6 to 12 hours to record baseline data. For the metabolic chamber study birds, the loggers only remained attached while the birds were inside the chamber, and the heart rate was recorded over 5-second intervals. The lowest average of measurements over a 1-hour period was considered to be the basal heart rate. The heart rate of the incubating birds was significantly lower than the birds in the metabolic chamber. For incubating birds, the average heart rate was 56.9 beats per minute, and for birds in the metabolic chamber, it was 93.1 beats per minute. This suggests that birds in the chamber were under stress, and that resulted in artificially high heart rate levels. The metabolic chamber was inside of a thermostatic box so that the temperature within the chamber could be maintained at 8 to 10 degrees Celsius, the same as outside air temperature. A vacuum pump pulled air through the chamber to help maintain temperature, while a subsample of the exhaust air from the chamber was continuously drawn through columns of dryrite. Dryrite is a drying agent for air and gases. It just trapped the moisture from the air. Oxygen content was measured by an oxygen analyzer and then drawn into an infrared analyzer to measure carbon dioxide content. The measurements were averaged for every 20-second interval and stored on a computer where software was used to convert changes in oxygen to values of rate of oxygen consumption in milliliters per minute. Then rate of oxygen consumption was converted to energy expenditure. Before any measurements were taken, the birds were allowed to get used to the chamber for a minimum of 1 hour. Measurements were made for heart rate and rate of oxygen consumption for 3 to 9 hours. The lowest measurements of rate of oxygen consumption, averaged over a period of 50 continuous seconds, were considered to be the basal levels. Although the digestive state of the birds went undetermined, past observations with non-breeding adults had shown that they do not eat while at the breeding colonies, so Weimer, Skirch, and Associates believed that the birds were in a post-absorptive state, a conditional requirement of basal metabolic rate. For birds in the chamber, rate of oxygen consumption was much higher for females than for males, in absolute and mass-specific terms. Even so, basal heart rates of the birds in the chamber did not differ significantly between male and female. The average basal metabolic rate for males was 138 kilojoules per kilogram per day, while the average basal metabolic rate for females was 230 kilojoules per kilogram per day. Rate of oxygen consumption was found to correlate significantly with heart rate, and the slopes for individual linear regressions were all similar, but the intercepts were not. The intercepts were different for each bird due to differences in size. The slope from the pooled data and the average intercept was used to predict rate of oxygen consumption from heart rate. Basal heart rate was positively correlated with mass and energy expenditure of incubating birds. We have essentially reviewed the first purpose of the study, which was to measure heart rate and rate of oxygen consumption to determine energy expenditure for both birds in the metabolic chamber and free-ranging birds to see how they differ. The next purpose was to measure effects of mass, age, and sex. Basal heart rate was similar for males and females. After being fitted with the recorders, it took an average of all birds about 120 minutes to return to basal levels. The males seemed to recover faster within the first 20 to 30 minutes, but then there was no real difference between males and females. You can look at the figure here to see the male and female heart rates returning to basal levels after handling. The 20-minute mark has an asterisk to draw attention to the steep difference in male and female heart rate at this time. The male and female heart rates returned to similar values at about 50 minutes after handling. Mass was significantly related to heart rate, so that heavier birds had a higher average heart rate, but age made no significant difference. You may recall that the third purpose was to determine the effects of weather and daily rhythms on the incubating wandering albatrosses. For incubating birds, heart rate varied throughout the day. The variation was so great that the confidence interval was only at 20.3%. From 8 o'clock a.m. to 1 o'clock p.m., the birds had the highest average heart rates, which were about 20% higher than the basal levels. This may have been due to increased interactions with conspecifics or predators such as skuas. At night, there was a decrease in heart rate to a range within 10% of the basal levels, probably due to decreased interactions with conspecifics and predators that caused a reduction of alertness, allowing the birds to sleep better and thereby lowering heart rate. Weimer, Skirch, and Associates warned that because the basal metabolic rate and heart rates varied so much between this one group of birds, that caution should be taken before comparisons of these measurements are made between different species. A weather station that recorded daily air temperature, wind direction, and wind chill was used to monitor field conditions of the birds at Point Baz. The only meteorological condition that affected heart rate was wind chill, but only for temperatures below 0 degrees Celsius. There was no effect for wind chill above 0 degrees Celsius or for wind air temperatures, precipitation, or other factors. The bird's heart rate increased in response to wind chill with temperatures below 0 degrees Celsius, possibly to compensate for the wind disturbing their plumage and reducing their thermal insulation. Compared with other subarctic areas, the Crozet Islands have milder temperatures. Weimer, Skirch, and Associates predict that in colder areas, albatrosses will have higher costs of incubation because of the effects of wind chill. If you look at the figure here on the slide, you can see the heart rate as a function of wind chill for temperatures below 0 degrees Celsius. Heart rate increases as the temperature goes further into the negatives. Other natural factors that affected heart rate were noted by Weimer, Skirch, and Associates. Birds that were brooding chicks had a higher heart rate than those that were incubating eggs. Although the researchers are not sure why, it may be possible that the cost of heating the chick may be higher than the cost of heating the egg. The final purpose of the study was to measure the effects of human disturbances on the albatrosses. For the Point Baz birds, effects of human disturbances on heart rate were measured when a human was various distances from the nest. The birds here had minimal interactions with humans. The only time they ever encountered humans was once or twice per breeding season for band controls. Therefore, they were not accustomed to human presence in the breeding colony. Because basal levels had been recorded for all birds, each bird was able to serve as its own control. To measure changes in heart rate for human presence, a human observer would approach the nest in a straight line during time intervals. When the bird noticed the human, the time and distance from the nest were recorded. Then the observer continued to move closer to the nest until it was 1 meter from the bird. Once the observer was at 1 meter, they moved away from the nest until out of the bird's view. A similar trial was done, except for when the observer got close to the the bird was captured by the beak so that its band could be read. The band control lasted for 10 to 20 seconds. Then, for 8 other birds, a second band control was done to measure any differences between the first and the second trial. As soon as the bird saw the observer, the heart rate sharply increased for both males and females to nearly twice the amount of the basal levels. The average heart rate continued to increase as the observer approached. Interestingly, males had a higher heart rate compared to females as the observer approached within 50 meters of the nest, but when the birds were captured for a band control, the heart rate between males and females peaked at similar values. When the birds were handled for the band control, the heart rate was about 4 times the basal levels. Because the male heart rate increased more quickly, this could mean that males are more prone to stress and are more likely to act out aggressively than females. Even though males had a higher heart rate when the observer was nearby, heart rate took the longest for females to return to basal levels. For males, heart rate took about 9 minutes to recover, but for females it took about 12 minutes. For 8 birds, a second band control was done between 5 and 18 hours after the first to see if they reacted as strongly as the first encounter, which they did. There was not any significant difference between the first and second band control for these birds. The heart rate before handling and the prolonged recovery period due to stress would have raised energy expenditure for the birds. Weimerskirch and Associates state that the stress involved is not negligible, and that for smaller species, the effects of stress could have an even greater impact on energy expenditure because energy stores decrease with body size. Other than these experimental trials with human presence, the birds had no additional human interaction, so for the remainder of the study, the birds experienced natural conditions. So far, we have learned that energy expenditure can be affected by several factors, such as basal metabolic rate, body size, meteorological conditions, presence of predators and conspecifics, presence of humans, and human disturbances such as handling. To measure the impacts of these disturbances and other factors that may affect energetic costs of free-ranging animals, we need to know the instantaneous measurements of energy expenditure in the field. Heart rate is a convenient way to do this. The loggers made it possible to get instantaneous measurements, whereas a doubly labeled water study would have averaged the energy expenditure over the measurement period. However, a doubly labeled study by Schaefer and Associates 2001 measured field metabolic rate of incubating wandering albatrosses, and the results were not significantly different for incubating birds used in this study, although they were somewhat higher. Even though respirometry is the most commonly used method for measuring metabolism in resting animals, it can make the animals stressed. This can cause the measurements to be overstated, as stress levels may be elevated from having to be inside of a chamber or wear a mask, or other things that might make the animal nervous. For example, a 1984 study by Brown and Adams measured energy expenditure of albatrosses using respirometry techniques, but the birds had to wear a mask over their bills. Brown and Adams noted that the males appeared agitated, so the birds were likely stressed. The energy expenditure that Brown and Adams measured was about 2.5 times higher than what was measured in this study for the free-ranging birds, which was 1.7 times lower than those measured in this study by the metabolic chamber. For these reasons, Weimerskirch and Associates believe that heart rate, incubation costs, and basal metabolic rate measurements from previous studies are overstated. Heart rate increased with the presence of humans and handling, and while in the chamber, birds maintained a higher heart rate, meaning that the basal metabolic rate was higher than the birds in the field. Rate of oxygen consumption measured would be higher, especially for studies that are only a few hours or less, as this may not even be enough time for the heart rate to return back to basal levels in some cases. Another advantage of the data loggers is that they can record heart rate for days, weeks, or months. Heart rate of free-ranging animals using data loggers can be used to estimate a lower energetic cost than other types of studies that may cause an animal to become stressed. The loggers can be used for a longer time frame than some study methods, allowing plenty of time for the animal's heart rate to return to basal levels. Basal metabolic rates and specific energy expenditure costs should be more accurate without the effects of human-induced stress, which, as we have seen, were the most disruptive, causing heart rate levels to be more than 4 times the basal heart rate in some cases.